As we noted earlier, nucleic acids are polymers, and they're composed of nucleotide monomers held together by linkages we call phosphodiester's. Let's get right to it with nucleotide structure on the next page. Nucleotides are in many ways simpler than amino acids, and one of the great things is there's only 5 of them instead of 20. They're also pretty simple to put together. They're composed of 3 things. First of all, we have what we call a nitrogenous base. That just means nitrogen containing base. It has a pentose sugar. That's going to be ribose or deoxyribose. And lastly, 1 to 3 phosphates. So let's take these one at a time. First of all, let's talk about the nitrogenous bases that we're going to encounter. I'm not going to ask you to draw these things from scratch. The structures shown below on the bottom of this page are always going to be provided to you in any exercise or exam that we have. You do have to recognize these structures and name them, but not draw them from scratch. So first of all, we need to get some nomenclature out of the way. There are 2 types of nitrogenous bases, and the first is called pyrimidines. And pyrimidines have a 6-membered ring with 2 nitrogens in them and 1 double bond. The nitrogens are spaced out as they are shown here, but again, you don't have to draw these from scratch. Purines, on the other hand, are 2-ring systems, and they have 4 nitrogens and several double bonds. The key thing we can say about these is that they are flat, they are aromatic, and they have an interesting dual nature about them. On the one hand, they are nonpolar because of that aromatic nature of the ring, but on the other hand, they have hydrogen bond donors and acceptors on the edges. And we'll point those out as we move on and look at the specific ones. Let's go below and take a look at the 5 bases that you need to know. Now, first of all, let me make sure you understand that there's a lot of names on this figure. The bases are named above each structure. So we have adenine and guanine for purines, and thymine, cytosine, and uracil for the pyrimidines. This is pretty much all we have to note about them, except that you do need to recognize them. And here's a little trick. The adenosine is the purine that doesn't have any oxygen in it. It's only got nitrogens, carbons, and hydrogens, whereas guanosine does have an oxygen in it. If you look down below, what we tend to do is think of thymine and uridine as the same thing. Why do I say that? Well, because they have the same core structure. And in fact, biologically, we almost treat them as the same thing. The only difference is that the thymine has a methyl group attached to it. In fact, it's the only base that has a carbon outside of the ring. So I hope these will help you a little bit in terms of recognizing them. At this point, it's time for us to build a nucleotide. We're going to do so on the next page. How do we do that? We're going to take a base, in this case adenine, and we're going to add a pentose to it. In fact, what we're going to do is add a ribose. Now remember, ribose is an aldopentose, and if we draw that in the Hayworth form, it's going to look like that. Let's pause here and see if you can do a little review from a previous chapter. I'd like you to number the carbons in the ribose, and I would like you to tell me where the anomeric carbon is and whether in this current state right now it's in the alpha or the beta form. Pause for a moment and take care of that business. Okay, I hope that you would recognize that carbon number 1 is right there, 2, 3, 4, and 5. Now what's interesting is that when we have a ribose, we number them such, but when we have an adenine attached to it, The adenine has its own numbering system, and so we use the numbering system 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime to indicate that those numbers apply to the sugar, not the base. Second of all, I hope you would recognize that the anomeric carbon is right here. Usually the rule is that it's attached to 2 oxygens, but in this case we've replaced the anomeric alcohol with the nitrogen from the adenine. And because of that, we can see that that nitrogen is above the ring on the same side as the high-numbered exocyclic, above, and that makes beta. This is a beta 1 prime N-glycosidic bond. We've seen nitrogens attached to sugars before, and we call them N-glycosidic bonds instead of O-glycosidic bonds. So let's acknowledge that at this point we have not made a nucleotide. Instead, we have made a nucleoside. And a nucleoside is a base plus a pentose. And in this particular example, we call this nucleoside adenosine. That is, if you take an adenine base and attach it to a ribose, it is no longer called adenine, it's called adenosine. And that's a name you need to know. In fact, that's probably more important than adenine. Let me just make a point. If we go back here, you're going to see that adenine is the name of the base, adenosine is below it. That's the name of the base once it's attached to a sugar. We've got guanosine, thymidine, cytidine, and uridine. You're expected to know those names. Now, that is a nucleoside. How do we make a nucleotide? Well, what we're going to do is attach a phosphate, and we're going to attach it up here. Sorry about that. You may not be able to erase, but you can draw right through that. And we're going to attach a single phosphate for now. And that's that. What do we call this linkage right here? Maybe pause for 30 seconds and ask, what is that linkage? Hopefully you recognize this linkage, that is COP double bond O, as a simple phosphoester. That phosphoester we learned back in our sugar chapter. We did it again in our lipid chapter. Here it is again, and we will be talking about phosphoesters for every chapter from now till the final exam. So, very important. Linkage of phosphoester at the 5' position. This now is called a nucleotide. And we're going to name this adenosine. We start with the nucleoside, and then we tag on 5' for the position of attachment, mono for 1 phosphate. You need to know how to name nucleotides and nucleosides, and this is how you do it. This is also called AMP, capital A, capital M, capital P for adenosine monophosphate. Let's have you try it. We're going to go over to the right-hand side of the page here. Here's a different nucleotide. Let's see if you can use the information on the previous page and come up with the name of this nucleotide. Okay, this one's a little trickier. The first thing we need to do is identify that the base is a thymine, and that is always seen because it's got that extra methyl group outside the ring. Now, if you go on the previous page, you will find at the core of it, thymines, when linked to other sugars, are going to be called thymidines. So we'll start with that. And let's take the easy part first. Because we have 2 phosphates attached, still attached at the 5' position, we are going to call this, I'm running out of room, so we'll put it down here, 5' diphosphate. No big deal there. However, there's another difference that we see in this nucleotide besides the ones we've already talked about. Do you see it? The answer is we're missing an alcohol here. There's just a hydrogen there instead. And because of that, we go back to our sugar nomenclature and say that this is called 2'-deoxythymidine 5'-diphosphate. So that is how you name nucleotides, and we'll practice that more on the homework. I'm past my 10-minute limit for video, so let's pause it here by just saying this would be called little d, capital T, DP. D, TDP for deoxythymidine diphosphate.